It is a great honor to be here, and I'm sorry I wasn't there in person. I wish I could be, but it's a great honor. So, is everything coming through sound-wise? All right, thumbs up. All right, so let me first say that I did have the wonderful opportunity to bring my family to New Zealand last year, and it was my first time there, and I was just blown away with just how beautiful it was. And we went North Island, South Island. And like I said, I can't wait to be back, but I appreciate the program committee for including me in this, and also thanks to the AV team for making this happen. In terms of the meeting, the content here, this is going to be a little technical. I'm going to try to keep it simple. I'm not the one doing the hard work here. This is my data scientists team and research engineers that are doing all the programs. So I'm more of the cartoonist, I guess, in this slide. But I am very passionate about kind of how we're going to use image-based data to hopefully help y'all treat your patients. And there's a lot that's unknown and a lot we have to figure out. But and hopefully I'm going to be able to show that there's some of what the questions are. But my goal today is to introduce what radiomics is to you. I'll present some of the results from 2 or 3 of our publications, and then the last part talk about what the limitations are and what we need to figure out before we can kind of adopt these clinical tools. We'll start off with some good news, just to kind of give this context. So we did a press release yesterday or 2 days ago to announce a 510(k) clearance in the US for a new scapular plate for these fractures after reverse. This will be able to come to New Zealand this year as well, which is great. But it's fantastic that Advita created a product for kind of a very small niche indication. But wouldn't it be better if we could kind of mitigate the complication altogether? And we've published some work on this, trying to identify patients that have certain risk factors for these fractures. And this is a 9,000 patient study that tried to stratify by age, gender, and diagnosis to figure out which patient cohort has the highest risk factor. And we were able to show 15 patients out of 9,000 that had an odds ratio of 10, and those were females over 80 that had RA. But it's not that complicated. You can't just use age and gender by itself, because females over 80 with CTA had a risk factor of 1.1, which is the same basically. So it's we got to do more in order to be able to prospectively predict these patients, and so I believe this is possible to use image-based data, radiomic measurements, to prospectively predict which patients are going to have these fractures, which patients are going to have glenoid loosening, which patients are going to have humeral loosening. And we'll kind of get into how this is going to be possible during this presentation. So what is radiomics? Basically, the premise is that medical images are clinical data. You just have to be able to have the computer process the data and quantify the data that's in there in order to understand what the data means. So that's effectively what radiomics is. It's using pixel level data that's numerically represented by the computer to give you more data that you can't see with your eyes. And there's a standard process that how you do that. So when you look at a CT scan, you have to segment and kind of just analyze one portion of it. So just the deltoid, or just the scapula, or just one of the cuff muscles. Then you have to process the image, especially if you're going to do multi-center data where you have different CT scanners, different acquisition protocols. To be able to kind of pull it all together and compare it and to the clinical data, you have to make sure they're all the same. And so there's a lot of processing involved. There's open source software that extracts this data using standard mathematical equations, which we'll talk about. And then you combine it with the clinical data, you build predictive models, you rank order the data that is being used to figure out what data is most important and what's relevant, and the data that's not being important, that is important, you just kind of ignore. So we'll start off with a little history lesson just to kind of make this interesting. I think the history is a little fascinating here. Sir Godfrey Hounsfield is one of the great biomedical engineers in history. He worked at a company called EMI in the UK, and in 1967, he invented the CT scanner. Interestingly, 10 years before, he actually invented the world's first all-transistor computer, which is kind of amazing. He was awarded the Nobel Prize. He was knighted. There's an interesting story about EMI. EMI, you may know, or maybe not. The M stands for music. They're a record label. In 1962, they discovered, or they signed a little band out of Liverpool called the Beatles. In '67, they signed Pink Floyd, and they kind of used the money from the music business to give Dr. Hounsfield a grant to come up with his CT scan. And he had this idea that X-rays were not quantifiable, but CT scans could be. And he created this absorption scale, which later got named after him to be the Hounsfield scale. And this is a screenshot from a paper in, I think it was 1980 in Science, where he basically says that. He talks about what the limitations of X-rays are, where it's just visual and subjective, and talked about the advantages of CT scans, where it's quantitative. You can quantitate what's there. Clinically, there's not really a lot of that. It's mainly used subjectively and visually, but that wasn't the intent. Really, radiomics is kind of manifesting what he intended all along. So what are radiomics? Well, again, it's math. It's available in open source software. It comes in different flavors. So you have what's called shape-based radiomics. You have first-order radiomics and second-order radiomics. Shape-based radiomics, what this is it looks at entire region of interest as a whole and looks at how curved it is, or the relationships between its long axis or its short axis. First-order stats are looking at the grayscale images within that region of interest and looking at just standard histogram stats. So it's like median, mode, range, interquartile range, that kind of thing. And then second order stats is pretty fascinating. It's actually looking at the relationship between pixels to each other. And so they talk about the spatial arrangements or the patterns of the pixels. And ultimately, the big take-home message here is it's just math. It has no clinical relevance at all. You have to combine it with clinical data, build predictive models, and figure out what has clinical meaning. And to kind of make that point, another kind of history lesson here, the most common second-order radiomic measurement is something called the gray level co-occurrence, or GLCM. It was first presented back in 1973 at this paper that was been cited 31,000 times. This isn't a medical paper. This is actually, it's kind of an industrial application. If you read this paper, they talk about trying to identify from satellite images and aerial images how much farmland is in like Monterey Bay, California, or different types of rock, so you can figure out where to drill for oil based on these photographs. And so again, to make this data work, to make these measurements work, you have to combine it with clinical data, because some of it isn't meaningful, but some of it is. And so to talk about kind of the relevance of it, muscle and bone, good bone looks different on an image than poor quality bone. Patients with a lot of fat in the rotator cuff look different than patients with less fat. You can see that in the image. So you should be able to mathematically represent that using some of these tools. And so, for example, like a patient that has a cyst in the glenoid, you should be able to use a gray level size zone matrix, which is the third one on the right, to identify whether or not that cyst is there or not. Problem is, we just have to make sure these tools are validated so it actually does what we want. But mathematically, it should work. It's been used very limitedly clinically. And where it's been used is in the oncology field. And actually, Dr. Hounsfield, what he originally developed a CT scan to do was to non-invasively biopsy or non-invasively view tumors in the head. So it was kind of a much smaller scanner. And I mean, he actually used it on himself. It just kind of let you know what kind of man he was. It's kind of a lot like the story with Dane Miller, founder of Biomet, implanting titanium to demonstrate how safe it was. So it's basically using these kind of shape-based measurements. They've been able to show that benign tumors are typically round, smooth, and compact. And you can use shape-based radiomics to identify those. Conversely, they've been able to show that malignant tumors are irregular shaped and kind of spiky. And you're able to identify those too. The other thing that I think is really fascinating and how they've been able to kind of build models to show this is that they can use those second order pixel measurements to look at the outside of the tumor in the image and infer from the phenotype, how it looks, what the genotype is. And so tumors, they change, and so depending on the genotype, they're able to figure out how to treat it pharmaceutically. So it really is truly precision medicine, but there's a lot of applications beyond radiomics. The thing that we really have to figure out is these measurements are very sensitive to a lot of different steps in the process. So image acquisition, pre and post processing, how good of a segmentation you do, all of those things affect how stable these measurements are. So when you read this literature and when you do this work, you have to process the images first to account for these different things. And when you read the literature, people will talk about resampling, which is just making sure all the images across the data set have the same voxel size and resolution. They talk about normalization, where you're kind of redoing the grayscale to a mean and deviation, so it kind of spreads the image out more evenly. And then from a compute standpoint, there's something called discretization that changes like a 256 grayscale image to, let's say, 40 bend. So it's 40 different shades. And as you can see in the pictogram, the histogram down at the bottom, it's meant to simplify it and make it easier to do these calculations. So with that as a background, let's talk about what the studies that we did are. And with any ML study, it's garbage in, garbage out, right? You got to use good data you trust. What my team is able to do, we're leveraging our clinical outcomes database that we have about almost 50 sites now, over 22,000 patients enrolled. Since the beginning, everybody's used the exact same forms to collect the exact same data the same way. The patient consent and the RBs that we have includes the CT scans. So because CT-based planning is so widespread, of those 22,000 patients, we have over 6,000 patients that have CT scans now. And so it's really a great opportunity that you can overlay the CT scans with the image data and do this work. So the three studies we're going to talk about, and I'm going to just kind of pull data from it to kind of tell a story. But I think we were the first to do a radiomic-based analysis in the shoulder, certainly the only to do it, a huge study. It was last year, we published a paper with 1,000 shoulders in it. Just last week, we had a paper that went online. It's still in press, but it's approved, that had a 4,000 patient analysis. And that's what we're going to talk about today. And there's another that's in process as well. All these papers have a very similar methods. Basically, step one is you auto segment the image. If you're going to do a 4,000 patient study, no one's manually segmenting all these. So you have to validate those algorithms first, and we've already done that. After you kind of isolate and create these regions of these 3D volumes, region of interest, you process those images and you build an ML framework to automatically do the math that's in the image. And so that's called extracting the radiomics. Then you overlay the radiomic data with the clinical data and you build the predictive models. And so we've built pre-op models and post-op models to predict outcome measures, which you can see there, PROMs. Our PROMs, obviously, active range of motion measures. And then we do it specifically at pre-op, 3 months, 6 months, 1 year, 2 to 3 years, and 3 to 5 years. And so then after you make those predictions, it's not a black box. What you're able to do is, with the algorithms we use, there's something called the F-score that allows you to rank order and figure out what data the computer used to make its predictions. And that's really helpful for radiomics because, again, the measurements itself have no clinical meaning. You have to figure out from the computer what data is most predictive. And then ultimately, we did a clustering analysis, and I'll explain why. I think it's pretty fascinating when we get to it. So some of the measurements, just to give you some examples, you can measure how flat or how curved a deltoid is, and this is the equation for it, the least, square root of the least over the major axis. You can measure the sphericity, how round it is, but it's more than that. It's also a measure of thickness of the deltoid because of the volume and the area equation there. There's different measures of volume, either a direct measurement and also a normalized volume relative to age and gender. We do lengths and width measurements and similar measurements from the scapula as well. Then we have fat percentage measurements, which are less reliable and probably we won't use going forward. In terms of the data, what I'm showing here really isn't that important. What's important is that we see lots of differences in these measurements, and some of the measurements are pretty tight. in terms of what the deviation is relative to the mean. So we see differences in age and gender, in prostheses type, in image kernels, which I'll talk about later as well, for both the deltoid and the scapula. But the big question is if you create a model and you have the computer make predictions and it assesses how accurate it was, and then you create a new model and give it radiomic data, is it more accurate? And the answer to that, when you give it deltoid radiomic data, the answer is yes. So about 70% of the predictions that we've done are improved by the deltoid radiomic data. It's not massively improved. You see like 5 or 10%, but it is improved, and it's only improved in overhead motion, which you'd expect with the deltoid. Same thing with the scapula. About 67% of the predictions that we did when you added in scapular radiomic data were improved, and some of the measurements were 10, 13% improved. So it's, they're already were pretty accurate, but this is just making it more accurate and more personalized. Another advantage is these are automated measurements, so you don't have to ask a patient or anyone else to input data. The computer is able to just get this information automatically, so it's more usable. So then we looked at what is driving these models, what data is most predictive, and what we've known before is that pre-op PROMs, pre-op range of motion, retroversion, beta angle, those measurements the computer loves for making predictions. It's highly predictive. There's some demographic data that's very predictive. And then just after that, that's where all the radiomic data comes in. First, shape-based measurements are most predictive, then first order, and then second order. And finally, the least predictive data is information about patient comorbidities, patient diagnosis, and actually, as an implant manufacturer, this is kind of painful, but implant size and type data is pretty much the least predictive. But don't hear that this, because it's kind of middle of the road prediction, don't hear that the radiomic data is not that important because it's the fifth one down. When you zone in and look at specific radiomic measurements, there's a bunch of measurements in the top 20. If you look at the top 40 things driving the pre-op model, 28 of them are radiomic data. You look at the top 50 driving the 2 to 3 year model, 28 of the 50 are radiomic data. And the deltoid makes up more of those relatively than the scapula does, but there's highly predictive data of both the deltoid and the scapula. So then one thing we did just from a validity standpoint is we looked at patients that had complications for, let's say, reverse patients that had stability, and looked at the radiomics of those that had it versus that didn't. And we also did the same thing for patients that had scapular fractures versus those that didn't. And there were a lot of radiomic measurements that showed significance. So that kind of gives confidence that we'll be able to kind of do what I promised at the beginning of the meeting in terms of building a predictive model using this data. So let's go into the limitations and kind of the challenges. So from a regulatory perspective, when we're building medical device tools that help healthcare professionals make decisions, it's really important that the user understands what the data is. There's a lot of requirements around that from a regulatory perspective, guidance documents, and that kind of thing. And radiomics are a challenge, right? Because you can't see pixel level data. You can't do these mathematical equations on a sheet of paper. You have to depend on the computer. And so that's going to be problematic. The other point is everyone's building these CT-based planning softwares. It's the hub for a lot of different decisions that are made prior to surgery. It's not really practical or usable from a UI UX standpoint to load a CT scan and get a 3D image of a muscle and bone and then give you 100 different radiomic measurements. That's just, you wouldn't do anything with that data. So there's another machine learning operation, a technique called clustering, which may be a solution for this. And I don't know if it will be, but I think it's pretty interesting. So what clustering is basically you give the raw data to the computer and it's able to figure out things that occur naturally together. And so kind of groupings. And what we did, and what's great about this is if we can identify clusters or groups, you can prospectively use this in the planning software. So, if we find a unique deltoid, you can actually prospectively identify in the planning software and tell you, hey, this is a deltoid grade B or C or whatever you want to call it. And so that's basically what we try to do. So we took the top 8 most predictive radiomic data points. We combined age and gender and we asked the computer to find groups. And then we overlaid, and so there's four male deltoids and four female deltoids. And you can see in this graph, hopefully it's coming through, kind of how the data is spread. There's some overlap, but they're definitely distinct. This is males, this is females. So then we overlaid that with the clinical data to see, are there differences in pre-op motion and proms, and also post-operative differences. And we do see that. There are some deltoids that do better prior to surgery. So you can look at the image and you can say, hey, this is the deltoid that is bigger, thicker, wider, whatever, and it's associated with more motion. You can also look at patients that have thinner, frailer, smaller deltoids, and they're associated with less motion. So it's from a validity standpoint, it's good that we're able to see in the data. And maybe some of these measurements and groupings of the data and of the muscles also speak to improvement capabilities. And so we're able to see an M2 deltoid, the green dot further to the right. That means it has more motion, more proms. This is for males at 2 years. You look at the female data. The F2 deltoid is further to the left. So that means it has less proms, less motion prior to surgery. And then at 2 years, females that have the F0 deltoid had more, significantly higher proms. So again, this is just kind of a feasibility analysis, but the whole idea of this is we're trying to put together very complex information and make it visible, and make sure that's clinically relevant relative to the measurements you take in clinic. You can do this for bones. You can also do this for subsets of bones. So if you wanted to look at different types of glenoid vaults, you could create, or different types of acromions or scapular spines, you could do this analysis and come up with different classes of morphological structures. So last minute or so, what I promised at the beginning about kind of a sub-volume assessment of the scapular spine, we haven't done that yet. We're still in process. This is looking at the entire bone or the entire muscle. We've built audio segmentation algorithms for the rotator cuff muscles, but we haven't finished validating it. So we haven't done that yet. That's coming. All of this is retrospective data, so inherently it's correlative, not causative. And there's a lot of questions we still have to figure out. So something you may hear about or may know, radiologists will use something called convolution kernels to bring out contrast in imaging. It's basically a filter. When you analyze images of different kernels, shape-based measurements are completely unaffected by that, but any grayscale-based measurement is affected. And so two of the most common kernels that we see are bone kernels and FC30 kernels. Those are two different CT scanner manufacturers. The average fat of a bone kernel patient is 10%. The average fat of a FC30 patient is 30% of the deltoid. So clearly it's the image, not the patient, right? So there's processing you have to do. And this is the exact same patient, the exact same slice with two different image filters. And one says it's a 7% fatty infiltration of the deltoid. One says it's 1.5%. So there's obviously questions about ground truth that we have to figure out. And we're doing those things from cadaver studies, work on that. Company that has 10% market share, you see a lot of different CT-based kernels, but for the most part, there's 5 or 6 kernels that are about 70% of the ones we see. So those are kind of the ones we're focusing on. There's questions of fat outside the muscle versus intramuscular fat. Does one matter more than the other? Should we exclude one? We're trying to figure all those things out. There's questions of, can you use these second order base radiomics to analyze and find patients that have fatty accumulation? Is fatty accumulation in one part of the muscle more clinically relevant than something that's more spread out evenly? All things we're trying to figure out. And then finally, metal throws off CT images, and it's not just metal in the joint. You can have metal on the other side of the body, and it'll still scatter and throw off these measurements. So you have to kind of exclude these patients and catch them. So you don't, if you're going to do this analysis, that doesn't throw off your data. And the last slide is that we have to make sure that this clustering analysis really is predictive, because we know from previous research that some of the classification systems used aren't really very predictive of clinical outcomes. Walsh, Favre, and Antonio, we've been able to show from ML models are basically on the bottom 10% of the feature importance data. And we have a couple of publications about just using other statistical analysis to show how unpredictable they are. So hopefully we can create predictive models in new ways. I think shoulder is going to lead all of orthopedics in this type of work because of the widespread use of CD planning. Exactech does have a predictive model software that is already image-based. So this is a screenshot of something that's currently available in New Zealand and Australia and Europe, where we're using image-based measurements to make predictions. And that's what Predict Plus is. So it's loading in measurements of automatically collected of retroversion, beta angle, subluxation, and image implant data. But ultimately, what we're trying to build is a third version of that that's based on radiomic data, which will have muscle information, bone information, not just the outside, but what's going on inside in the pixel. So this is one more thing my company is working on that I think is going to be very differentiated, and we're probably a year and a half away or so from having something that's available clinically. A lot of regulatory work still to do as well, but again, really optimistic about it, and I'm now happy to take questions.